So for now, we're going to move on to Kristen Marie Westfall, who will be discussing applied molecular biosurveillance in the Northeast Pacific, Part 2, improving targeted detection and quantification. So this is going to be a continuation of Tom's talk.
Thank you, Jeanette. I just want to check that everybody can see my screen in presentation
Yes.
mode. All right, great. So yeah, so my talk is a continuation of applied molecular biosurveillance. And this talk is actually going to focus a bit more into the targeted detection and quantification side of eDNA. So the driving force, the ultimate goal behind this research was to implement eDNA tools into invasive species management. And two of the main objectives to reach that goal were to develop new eDNA tools to meet the current management requirements and to understand how eDNA behaves with respect to traditional survey metrics. And Tom just presented the development of one new eDNA tool in our lab and how the data that we gathered from this tool, how we compared it to data from settlement plates. And I'm going to describe the development of another new eDNA tool as well in this talk. So I won't go into background about non-Indigenous species or eDNA. I think it's been very well covered. And I'm going to basically just go straight into targeted NIS detection, describing a case study of the European green crab. I'll give you a bit of background about green crab in North America, and then go into a new quantitative eDNA-based assay that we developed that we've called targeted next generation sequencing, and how we use this assay to quantify the relationship between traditional survey CPUE, or catch per unit effort of green crab, and eDNA availability. And we also compared eDNA detection probability and the power to detect changes in crab abundance between two quantitative techniques. That is the new TNGS assay that we developed and qPCR. I'm going to bypass this slide entirely because we all have a very good grounding in eDNA right now, I think. So the European green crab, Carcinus manus, came to North America in the 1980s. It established itself in coastal regions in Washington State and the west coast of Vancouver Island during the late 90s, and it's been actively monitored by DFO since 2004. The green crab is considered one of the world's 100 worst invasive species. It's a voracious predator. It can decimate fragile eelgrass beds. In all, they exert considerable pressure on the native biota and have extreme impacts. So because of the extreme nature of the species, there have been major efforts in Canada and the US to monitor their distribution and eradicate nascent populations as they arise. And this is particular to the Salish Sea, which includes Puget Sound, Strait of Georgia, Juan de Fuca Strait, and other smaller regions in that area. There are small, low-density populations that have cropped up in the Salish Sea, and we've been able to detect them, or agencies have been able to detect these fairly quickly and eradicate them. But there is considerable potential for eDNA to monitor the spread of populations, quantify their abundance, and also assess eradication efforts. So the targeted next generation sequencing assay that we developed is based on the qPCR assay, or based on the qPCR technology. eDNA is amplified from the same primers as developed under the MIQE guidelines for qPCR, but products from a single-step PCR reaction are sequenced at equal volume on a high throughput platform. In our studies, we use the Illumina MiSeq. And For those who are into the technical side of processing eDNA samples, this is done, the PCR products are added at equal concentration, or equal volumes, I should say, without normalization of concentrations. TNGS offers some benefits over qPCR. First, it circumvents the limitations of fluorescence-based assays for DNA at very low concentrations. The qPCR assay that was developed also in our lab for green crab was able to detect DNA at the lowest concentration of 2 copies per microliter. And that was in all of the replicates. And that number dropped down to 0.2 copies per microliter if we considered just the 50% of the replicates. However, the TNGS assay was able to detect DNA at 0.02 copies per microliter. So this is a 10 to 100 times decrease in the limit of detection, indicating that this new assay has the potential to be more sensitive than qPCR. And we'll show that this potential was realized in our field surveys. But just like in qPCR assays, standard curves are generated during each sequencing run to extrapolate eDNA concentration from unknown samples. This is a sample standard curve that we obtained from one of the MySeq runs. We had 2 MySeq runs in this study, and both of them had R-squared values around 0.96. So our case study on detection and quantification of invasive green crab was concentrated in Barkley Sound, which is shown on this map. This is on the west coast of Vancouver Island. So here, green crab populations are extremely patchy. They range from small, very low abundant populations in the east to very highly abundant, quite dense populations in the west. We trapped green crab and collected water samples for eDNA analysis at 2 low abundant sites and 2 high abundant sites. And the objectives here were to define the relationship between eDNA concentration and catch per unit effort, or CPUE. We then compared the performance of the TNGS and qPCR assays with respect to the probability of detecting crab at low abundance, and also the power of each assay to detect changes in eDNA concentration, and as a proxy, crab abundance. Oops. Sorry. So figure 1 on the left here shows a positive relationship between eDNA availability and CPUE for both assays. But what stands out here is that the linear model explains more variation in the TNGS dataset than it does for qPCR. And the predicted CPUE values are more accurate due to the smaller 95% confidence intervals in the TNGS data. And this is especially apparent at the low abundance sites. The box plots in figure 2 show a similar relationship. The TNGS shows a clear difference in eDNA concentration between the 2 low abundant sites and the 2 high abundant sites. But the difference is not as clear with qPCR. The high abundant site here with a CPUE of 23 showed a similar eDNA concentration as these 2 sites, CPUEs of 2 and 7. So we built a hierarchical multi-scale eDNA occupancy model in order to estimate the conditional probability of occurrence of green crab eDNA in a sample. As expected, the probability of eDNA occurrence increases as crab abundance increases. The detection probabilities at the high abundance sites were similar between the 2 methods. They were ranged between 95 and 99% at the high abundance sites. But the TNGS assay on the top here shows slightly higher detection probability at the low abundant sites. And this relates back to the lower limit of detection I was talking about that we got from the standard curves. And this translates to a 7 to 10% increase in detection probability at these 2 low abundant sites. This may seem like a very marginal number, but this is potentially valuable when searching for very low density populations, like during secondary spread, have the nascent populations that are just a few crabs probably. So one of the most challenging aspects of quantitative eDNA research is the intrinsic spatiotemporal variability of eDNA in aquatic environments. We looked at which parameters were causing variation in concentrations in these two data sets. And here's a plot that partitions the sources of variability in both data sets. In the biological category on the left shows the variation among 6 site replicates that were taken within each sampling day. The within-site variation, WS, adds the variation that's contributed from the qPCR technical replicates. We couldn't calculate the same value for the TNGS because each of the technical replicates for this assay had the same molecular ID. The days category shows the variation within sites between 2 sampling days that were taken about 24 hours apart to capture variation introduced by tidal cycles. And it's important to see that the variation among days is very similar to the variation within days. It shows us that eDNA is fairly endogenous to each site, doesn't change appreciably over a tidal cycle. The last points on the right show the variation between sites, or the variation in eDNA concentration across Barkley Sound. We can think of it that way as well. The difference from between site to within site variation is greater for the TNGS assay than it is for qPCR, which shows us that TNGS has more power to detect changes in eDNA concentration and by proxy detect changes in crab abundance. So I want to highlight some of the take-home messages from this body of research. Here we've improved green crab detection and quantification using eDNA by carefully considering specific management needs. And importantly, we have demonstrated that both quantitative assays, the qPCR and the TNGS, are useful for monitoring green crab. So by demonstrating that CPUE and eDNA concentration vary similarly, we can now use eDNA for estimating crab abundance. And we can also use TNGS as a highly sensitive method for detecting green crab at low abundances, such as searching for nascent populations in the Salish Sea. This leads to the question of where do we go from here? Earlier in the session, we had a talk about digital droplet PCR, and it would be a very interesting study to compare all 3 methods with the same data sets. And we also need to standardize methods, including the field sampling, laboratory analysis, bioinformatics analysis, and the all-important reference libraries that have been a great theme. And when these methods have been standardized, we can and should implement them into our regular monitoring programs. So we have the same acknowledgements as the previous talk, so thank you to our funding sources and collaborators. And if there are any questions.
Yes, thank you, Kristen, for that talk. And we do have a question. We have a question from Hitoshi. He says, doesn't the sensitivity of the TNGS method depend on the targeted number of reads per sample? If so, what is your recommendation for a reasonable number of reads for avoiding false negatives under low abundance of target species? So for an example, 50,000 reads per sample.
So let me just, here we go. I just want to read the question here. Here we go. The targeted number of reads, recommendation for reasonable number of reads for avoiding false negatives under low abundance. So we had our lowest limit for detecting species was actually 100 reads. And there are instances, this is part of ongoing research, because there are instances of tag jumping and being able to quantify the prevalence of these artifacts, these high throughput sequencing artifacts, is part of ongoing research. But because we were able to see clear patterns in the data with little variability, a small degree of variance between replicates, and also we found the strong positive correlations with CPUE. So the influence of false negatives is likely to be quite low. Are there any more questions?
Yeah, I don't see anything else more in the chat.
Christine, can I ask you a question directly about my previous question?
Yes.
This is not about the artifact or anything. It is about it's a targeted number of reads per sample. You can use the whole run for one sample if you like.
Right.
So the huge number of reads can be applied to a single sample instead of hundreds of samples as usual.
Right. Yes.
But
we were multiplexing about 30 samples per MySeq run.
So that's a allocation of very high number of reads per sample then.
Right. Yeah. So if you sorry, I can't. Let me share my screen again here. If we go back to the standard curve here, this is the raw number of reads that we were dealing with for each, for the concentrations of DNA in the standard curve. So we're looking at very large numbers of reads. It wasn't based on, say, 5 or 10 reads per sample.
Right.
Yeah.
Okay, thank you.
And because it's a very new technique, there's a lot of further research that can go into refining multiplex numbers and also looking at it in different systems. I'd really like to use, this was just one qPCR system that we borrowed from. So I'd really like to look at it in other species because there's so much variability in qPCR assays. And that was a highlight of the other talk, and also another reason to compare it with the ddPCR as well.
Okay, thank you very much.
So we have time for just one quick question. Here in the chat from Natasha, she just wants to double check. Did you see any correlation between CPUE and any measure of read abundance?
Yes, we did. So we back calculated the eDNA concentration based on read abundance. So in a double calculation way, the CPUE was originally related to read abundance, but I decided to change that into eDNA concentration just to be able to compare it with a lot of the qPCR studies out there that report, instead of reporting CT values, qPCR studies generally report eDNA concentration. That's been calculated from the standard curve.